guys, so it's Joseph here and welcome to another lesson. So in today's lesson I'm going to cover mitosis, explain everything that you need to be familiar with at your level. I'll cover what they frequently like bringing on tests and exam. Okay, but I'm not going to go deeper by a teach beyond the scope of your syllabus, because if you go deeper on this topic there are quite a lot of concepts. Okay, and unless maybe you later further your studies, maybe if you're going to study any medical program or pure biology, you're going to learn this in depth. Okay, just focus on what they ask at your level and what they really like to concentrate on. Okay, so what is mitosis? Maybe in case you are wondering, or maybe you're hearing this for the first time. So mitosis is simply one of the processes that a cell has to undergo in order to divide. Okay, one of the processes through which this cell undergoes cell division. Now, which kind of cell here? Remember, there are two types of cells. The main categories. The first one is the prokaryotic cells, and the second one is the eukaryotic cell. So now, for this mitosis here, we are focusing on the eukaryotic cells. Okay, we are focusing on the eukaryotic cells. I've already talked about the prokaryotic cells. For the prokaryotic cells, for them to divide, they undergo what is called binary fission. There is a separate video that I've talked about that. You find them in my tutorial group. To separate my tutorial group, there are like a bunch of lessons there. You find all the video lessons. Okay, so I've already taught that, but that's again not part of this topic or today's lesson. For today's lesson, we're just focusing on the mitosis. I'm just explaining so that you can just be familiar with everything. Okay, so for the prokaryotic cells, I've said they undergo binary fission, right? Now, for eukaryotic cells, for them to divide, now division it is the same way we consider it when you are looking at from a realistic perspective. For instance, let's say you have a chalk A. Okay, imagine this is a chalk. Then you decide to okay, let me divide it into two. How can you do that? You are going to break it here from the middle, right? What will happen if you break a chalk A? It is going to be splitted, right? There is going to be now two chalks. Okay, they are all coming from what was initially a single chalk. The same thing happens to the eukaryotic cells, or all the cells in general, though we are focusing on the eukaryotic cell for this lesson. So even for the cells, the same thing happens. If you remember when I talked about the cell theory, the postulates, there is a postulate that states that all the cells come from the already existing cells. Now, how does that happen? It happens by cell division. Okay, it happens by cell division. So initially there is just a single cell. Okay, the same way here there was just a single chalk. Now for even these cells, they have to split. Okay, they have to split at somehow the middle. Okay, and then when they split, they are going to end up with two cells which are identical to the original cell. Okay, now when you are dealing with, or when you are referring to cells, the one that was initially alone is referred to as the parent cell. This is referred to as the parent cell. And then this one here is, these two now that are produced as a result of the splitting of this cell here, these two are called the daughter cells. The daughter cells. Okay, now they might ask you, they might not ask you, but for just learning purposes, the reason why this one is called the parent cell is because it's the one that is able to produce these two, right? The same way parents are able to produce children, right? Now, for these two, they are called, why do you think they aren't called instead of being called, why are they called daughters instead of being called sons? Okay, they are called daughter cells because when a daughter cell is produced, even this cell again, it can also undergo cell division and produce two more cells. Okay, even this one, it is also able to produce two more cells. The same thing applies to daughters. Let's say you happen to have a daughter. That daughter will be able to produce two more. Us men, we don't give birth. Okay, it's not us, but the daughters are able to give birth, or the females are able to give birth. If a female gives birth to a female, that female is also able to produce another child, right? The same thing happens here. When this cell A, which is just alone, when it produces this cell again, it can also produce more cells. Okay, so this is what happens. In case maybe it is not making sense to you. So remember, I said that mitosis is just one of the ways in which the cells divide, right? Now, the other way is called meiosis. So this is another way in which the cells, in specific, the eukaryotic cells produce other cells or undergoes cell division or divides. Okay, so there are two ways. The first one is mitosis, and the second one is meiosis. Now, for this one, I'll just it will be another video. They just other concept on it, though they are similar to this one, but there's just some other additional concept on it. So it just needs another video. Let's proceed. Now, having explained this, now let's look at how now the mitosis process or cycle occur. And then one more thing before I even go any further. What you're seeing in mitosis, in some other books, it is called the cell cycle. It is called the cell cycle. When something is called a cycle, it means that it is repeating itself over and over again. Meaning that when it starts, let's say it starts from A, then it is ending from A. When it reaches the end or the last point, it is going to start again. Okay? Moving or revolving around the same thing, the same procedures. Okay? It doesn't stop. It's always happening. Then here comes after the metaphase, there is what is called the anaphase. So these are the 4 stages that the cell undergoes in order for it to complete the cycle of mitosis. And then the last one is called the telophase. So these are the 5 stages. So here's the thing now. Often they might maybe tell you to explain the cycle, or maybe they might just tell you to just explain a certain stage. Okay, so you just need to master all these 5 stages, because you don't know what they might ask you about. So now, the key thing here is to just be able to know what happens at this point, or at this stage. Now, you need to know them in order in which they are. Okay, you need to know starting from the one that comes first up to the one that comes last in the same order in which they are. There is no such thing as no. Maybe this time interphase happens before metaphase. No, and I'll share a secret with you. A mnemonic that I used to use that used to help me remember, that used to help me memorize these 4 stages. Okay, I was in first year at NS. And I'll share it with you so that it can at least make your life very easy, instead of having to remember how these. In case maybe you don't know, a mnemonic is simply a sentence, it's just a random sentence that helps you to memorize some sort of information. It's some information that is necessarily need to be remembered. Now, here is a mnemonic. So the mnemonic goes like this. It goes like this. I prefer making amazing tea. Okay, so let me just break it down for you. So this IA, the I is representing I mean, the I that the first letter A is representing the first stage, which is the interphase. Which is the interphase. And then the second word, as you can see, the first letter is P. So this P represents the prophase. And the M here, the first letter, is representing the metaphase. And the A here is representing the anaphase. Okay, the anaphase. And then the T here is representing the telophase. Okay, so I usually make a lot of mnemonics like this to help just my students remember what they are studying or what they're teaching, especially in chemistry and other courses that requires things that need to be memorized. Okay, so make sure that you join my tutorial group. You have access to simplified and explained videos like this. Okay, so this will make your life very easy to remember. Okay, trust me, guys. It happens that maybe you know what happens on each stage. Now you just don't know what is coming first. It might happen that they have told you maybe to explain what happens after the prophase. Now you don't know what is maybe the name of the stage that comes after. Maybe you might be remembering them in a disorganized manner. So you might instead maybe end up explaining the different stage instead of explaining the actual stage. So this mnemonic will really help you a lot. Okay, so now let us proceed. So having explained all these, let's look at the interphase. Okay, let's look at the interphase. So the interphase is a stage on its own. So it has some sub-stages or some stages that falls under it. The first one is called the G0. So the G0 stage, sometimes it is referred to as let me just write it down, then I pronounce it for you. It is pronounced as the quiescent stage. So this word, when something is quiescent, it means that it is at rest. Okay, so this is a stage where the cell goes at rest. Now, why does it rest? It rests because it's lacking some resources, for instance, maybe some nutrients that it needs in order to proceed on the next stage. A question I think once came, they asked about the importance of this stage. So this stage actually, oftenly, let me just say, it is when a cell is coming from a different cycle. When it has completed a certain cycle, then it has just restarted again. Okay, so it won't have any nutrients, like most of the nutrients that are needed for this whole process being used up. Okay, so when it goes here at rest, it is obtaining some nutrients. Okay, so it needs to procure some nutrients so that it can now proceed on on the next stage, which I'll talk about. So I think let me call this as 1. Okay, so that's the importance of this stage. Okay, because it needs some nutrients in order to proceed on the next stage. So let me call this A. Now, after it has rested, it will now go to the next stage, which is the G1 stage. So now, at this stage, the cell increases in size. Or let me just say, this is where it undergoes growth. Growth is where it increases in size. Now, they might ask you, what's the purpose of this stage? So this stage, the cell increases in size, or it undergoes growth, so that it can accommodate some of the materials that are going to be replicated. Because from this stage, it will undergo the next stage that requires the replication. to replicate is where you make a copy of something. Okay, let's say you have maybe a copy of an item here, then you make another copy. You have two, right? Now imagine you are making a copy of a lot of items, meaning that the number is going to increase. So now the cell need to accommodate. Now I'm just giving an example, but what replicates are called the genetic materials. The genetic materials replicates together with the chromosomes. Okay, so since they are going to replicate, it need to increase in size. Okay, because if it is small, then the materials that replicates are a lot. Maybe it might burst, right? So it need to increase in size. That's actually the main purpose of this stage. And then the third stage A is, let me call it our stage C. So this stage, sometimes often in books you find that they are calling it the S phase. So this S here represents the word synthesis, written like this. So synthesis, the synthesis of something is like to make something. Okay, the manufacturing of something. When you synthesize something, it's like you make something. Now, at this stage, there are quite let me just say, from the bigger picture, like two things that happens. There are some genetic materials that are being made, but one of the major things that happens is the replication of the DNA. Now, the reason why it is said to be synthesis is because as the DNA is replicating, remember DNA is made up of genetic materials, right? So first, more genetic materials have to be made, then the DNA now replicates. Okay. So now remember again, the DNA is found in, is contained in the chromosomes, right? So again, it's at this stage where even the chromosomes replicates. Okay, so let me just say the key or the main things that happen is the DNA replication and the chromosomes replication. Okay, so let me just draw a sketch diagram for the chromosomes. So what happens is like this. So initially, in the body of a prokaryotic organism, you as a single person, you are made up of, okay, so you're made up of cells that have 23 chromosomes. Okay, so there are 23. So now, each chromosome is going to be replicated. Remember, I said that when you replicate, it's like you make a copy of it. So another copy of this one is going to be formed. It is exactly as same as this one here. Okay, it is going to be formed, and this other chromosome that is formed is called the sister chromatide. So when they are replicated, so first the DNA replication. So you should mention DNA replication. After it replicates, since it is found inside the chromosome, then the chromosome now is going to be like another copy of it. It doesn't divide. It gets copied instead of division. The here is different. Okay, so another copy of this one is going now to be formed. So after now they are formed like this, they are now attached to each other. Okay, so they are kind of, just one more thing. Let me not forget. Here at the G1 phase or the G1 stage, this is where what is called the centrioles. There are some structures called the centrioles. The centrioles, they are formed at this stage. Okay, this is where they are formed. In case you don't know, centrioles are what bring about the formation of the spindle fibers. They are what are called the spindle fibers. They intervene at the stage called the metaphase, they attach to the chromosomes. Don't worry, I'll explain that. But for now, just know that these centrioles are what forms or are what result in the formation of the spindle fibers, and this is where they are formed. Okay, in case maybe they ask you. Now let's proceed on another. The last sub stage under interphase. Okay, so the last stage. Let me call this stage. Let me call it D. I'll call this D. So this stage is this is where now the organelles that are found inside the prokaryotic cells are going to duplicate so that they can be a lot of them. By organelles, I mean the structures. For instance, the endoplasmic reticulum. Let me write it down. The endoplasmic reticulum, the mitochondria, the Golgi apparatus. These are just examples. Let me just say etc. Okay, so let me just explain to you why this happens. So if you remember, let's say this is a eukaryotic cell, and then you know that it has got some organelles. Okay, let's say maybe these are organelles. Okay, now remember that it's about to be split. Okay, it's about to form two cells. So these have to be a lot. Okay, they have to be a lot. They have to increase in numbers so that they can be equally shared in the two cells that are going to be formed. Okay, so that's why they are going to duplicate. So let's say, for instance, this is a mitochondrion A, right? If it duplicates, there are going to be two, so that this one can go in this one, and the other one can go in the other daughter cell that is going to be formed. Even for the let's say maybe this is a Golgi apparatus A, okay. So meaning that initially in the cell, the parent cell, there is just one. Let's say there's just one Golgi apparatus. I'm just giving an example. Now, it needs to be shared in the two cells that are going to be formed. So it needs to be duplicated or replicated. To duplicate and replicate are just similar words. Okay, they are one and the same. So, and they say that when you duplicate, you make a copy of it. So now, imagine initially there's just a single, maybe say a single endoplasmic reticulum. Now, for the resulting daughter cells to also have an endoplasmic reticulum, or for each to have an endoplasmic reticulum, this has to be duplicated. Another copy of this one is going to be formed, meaning that now they are going to be two. One will go in the one daughter cell, and the other one in another daughter cell. So it is at this stage now where the organelles, the organs, or the structures that are found in the eukaryotic cells are going to duplicate. They are going to increase in numbers. Okay, so it is at this stage. That's the main thing that happens, and this is now to prepare the cell for mitosis. Okay, or for cell division. So in short, this interphase is just like the preparation stage. It's just for preparing now the cell to now proceed with the division. Okay, so now when it reaches at this point, already the DNA has been replicated, all the nutrients have been obtained. It is now going to proceed now in the cell division. Now this is where the stage of pure freeze comes in. Let me call this 2. Okay, so now what happens at this stage? What really happens? So now here there is a cell. Imagine this is a cell. It has got some organelles have been already replicated. So now remember, a eukaryotic cell has got what is called the nucleus. Okay, when you look at the structure of the nucleus, this is a nucleus, right? It has got this outer membrane here. This is called the nuclear membrane. or it is called the the nuclear envelope. The nuclear envelope. Okay, so sometimes you might hear them use the word nuclear membrane or nuclear envelope. Just know that they are talking about one and the same thing. So this is the nucleus. This is how it looks like, even though sometimes they just like shading it. So the nucleus, it has got here, it looks like structured. This one here, which is in nucleolus. Nucleolus. Okay, and then this space now here inside this O area, is what is called, it contains some fluid. It contains some fluid called the nucleoplasm. The nucleoplasm. Let me write it down here. The nucleoplasm. So here these are some fluid called the nucleoplasm, and then in this fluid is where now you're going to find the chromosomes. Okay, is where you're going to find the chromosomes which have crossed. They have crossed at the synthesis stage when they duplicated. Okay, so inside is where now the chromosomes are. So behind the hood, it is looking like this. Okay, and then remember I said that at the G1 phase, that's where there are structures called the centrioles. The centrioles. So the centrioles, when they are initially formed at the G1 phase, they kind of like looks like, imagine this is a cell, right? They are two centrioles that are eventually formed. So at first a single one is formed, then it is duplicated. Okay, meaning that at this point there are going to be two centrioles. Okay, there is going to be two centrioles. Right now I'm just trying to show you like the most important structures that are that plays a huge role at each stage. Okay, I know there are quite a lot of structures. The mitochondrion, the endoplasmic reticulum, and a lot of stuff. But I'm just trying to focus on what plays a certain role at each stage. Okay, so the centrioles, when they duplicate, they are going to move in the opposite direction. Okay, meaning that this one will go on this side and this one on the other side. So they will eventually going to look like this. They are going to look like this: one centriole A and one centriole A. Okay. So the centrioles, they duplicates at this stage. Initially there is just one. Then at this stage they are going to be replicated, meaning another copy is going to be formed like this, and then they move in the opposite direction. Now after they move in the opposite direction, so now these centrioles that you're seeing right now, they produce what are called the microtubules. Okay, let me write them down here. The microtubules. So these microtubules, they're like thread-like structured. Okay, very tiny thread-like structured. So it is going to produce the microtubules, which are going to join now together. Like many microtubules are going to join together and end up forming what is called the spindle fibers. So when the microtubules actually they even they are combined with some proteins, okay, but the microtubules are joined together like a bunch of them, so that it can eventually form the spindle fibers, which are even strong, okay, which are strong enough, because they need to be strong enough, because eventually they will need to separate the. Remember, at the moment, inside the this is a nucleus actually. This is a nucleus. So here inside the nucleus, there are some chromosomes that are crossed. Okay, so they are attached like this. So these need to be very strong so that they can be able to separate the chromosomes. Okay, there will be a stage where they will be required to separate these chromosomes. So they need to like combine a lot of microtubules so that they can eventually form something that is quite strong, which is now called the spindle fiber. Okay, so the spindle fibers are going to be formed. Right now, they're just going to be formed. Okay, they are going to be formed like this, but they won't be attached to any organelle or any structures that are involved right now. They're just here hanging inside the cell. Okay, so it is at this stage where they are now formed. Another thing here, these spindle fibers now that are formed, the combination of the spindle fibers, all the microtubules and the centrioles is what is called the mitotic apparatus. These are called the mitotic apparatus. So they might maybe ask you, let me say mitotic apparatus. So maybe they might ask you like, at which stage are the mitotic apparatus formed? Just know that they are talking about the spindle fibers. Okay. So another thing now that happens is that as these chromosomes are here within the nucleus in the nucleoplasm, as I've just showed you. Remember that they will eventually need to be make some sort of attachment with the spindle fibers that are just formed here, right? So eventually, when they are at the synthesis stage, when they're just being replicated, they are very small. Okay, they are very small that these can't even attach properly. So there's what is called condensation or the coiling. Okay, so they are going to coil more. They are going when they are coiled. It means that they are going to wrap around the. Remember that the chromosomes are made up of the DNA and the histones, right? Okay, so actually the chromosome is just a thread-like structure which just coils around the DNA and the histones. So it is going now to coil more so that it can become very thick, and as a result it is going to be short. It becomes short because it coils more. So it coiling more, what makes it become very thick. Now, when they become very thick, let me write here, they become very thick. So when it becomes very thick, it will even be more visible, okay? Meaning that these spindle fibers, they can now attach to them properly. I don't mean that at this stage they attach, no, but that's the purpose of the condensation that happens at this stage. Okay, so at the moment, when the cell just enters this stage, this nucleoplasm or the nuclear envelope, this one here, it is just sealed. Don't be confused. This is called the nuclear. This layer out A, it is the nuclear membrane. This one A is called. This is the cell membrane. Okay, don't be confused. They are different things. This is the cell membrane, also called the plasma membrane, but this is called the nuclear membrane. I know some students usually get confused, so I'm just trying to clarify it to you. So when it just enters this nucleoplasm, it is just sealed. Okay, so these chromosomes that are here inside, they need to come out so that the process can begin. Now, how are they going to come out from this nucleus that you're seeing here? So for them to come out, this has to break. Okay, the nuclear membrane, it has to break because they can't penetrate. They can't protrude through the nuclear membrane. No. So the nuclear membrane itself, it is going to break. Okay, it is going to be broken down. So the nuclear membrane breaks down. When it breaks down, it is going to look like this. Okay, it is going to look like this. It is going to have some sort of gaps. It is broken. Now the chromosomes can freely go out. They can now go out. Okay, they can now go out in the cytoplasm. This is actually cytoplasm. Okay, so that's all the important things that happens at this stage. Okay, so very important. sure that you watch this video over and over again if you're not understanding. I'm teaching is like everything that you need to know about mitosis. So now, at the end of this prophase, you're going to have this. So you're going to end up with a cell that is having now structures that are looking like this. So you're going to have the nucleus having the nuclear envelope that has been that has been broken down, okay, and the centrioles have moved in the opposite direction, okay. They have even formed the spindle fibers, and then even some chromosomes have gone out. They are just within the cytoplasm. This is the cell that you're going to end up with at this stage. Now let's move on to the third stage. So now at the third stage, this third stage here is called the metaphase. Remember the mnemonic that I showed you. Make sure that you use it very wisely. So I call this the metaphase. So now, what happens at the metaphase? So at the metaphase now, initially a cell will just come from the previous stage, that is the prophase, right? It will be looking like this. They are just the spindle fibers here, in the chromosomes. Okay, right now they're just scattered. Okay, they just disorganized. Now, at this stage, it is at this stage where now the spindle fibers, in some books they will just say the microtubule. Just know that they are talking about the combination of the microtubule that eventually form the spindle fibers. So if you want, you can say the microtubule attaches. You're just still referring to one and the same thing. Okay, so it is at this point now where each spindle fiber is going to attach to each pair of chromosomes. Okay, so imagine there is a pair like this. It is going to attach A at the center A. This center at their their region called the centromere. Now, in some books, they'll say that the spindle fibers attaches the pair of chromosomes at what is called the let me just write it down before I pronounce it for you. So they pronounce this word as kinetokyo. Okay, kinetokyo. That's how they pronounce this word. At their kinetokyo, meaning at their center A. Okay, at the center where they are joined together, where two chromosomes are joined together. So it is called the kinetokyo. That's how they pronounce the word. I don't know why they do, but it's just how they pronounce the word. Okay, so if you want, you can say the spindle fibers attaches the pair of chromosomes at their centromere or at the kinetokyo, but just know how to write it, since you won't be required to pronounce it. I'm just pronouncing it because I'm teaching you right now for just teaching purposes. Okay, so when now the spindle fibers attaches the pair of chromosomes at this point, it is going to attach like all the chromosomes are going to be attached. All the chromosomes are going to be attached, and when they attach, they are going to now like pull them in the opposite direction, since they are in their opposite direction, and then eventually align them in this manner. Let me show you. So this is the centrioles, with each centriole having the spindle fibers. So when they are going to be now arranged like this at the center. Okay, they're going to be arranged like this at the center. Okay, so at first the spindle fibers, look, there are some spindle fibers that are on this side. Okay, and there are some that are on this side. So the one from this side is going to attach, and another one from this side is going to attach, and then pull them now. So after pulling them, they are eventually going to end up being arranged like this. Okay, they are going to be arranged like this. Now, at this region where they are arranged in a straight line is what is called the metaphase plate. The metaphase plate, which basically just divide the cell into two equal parts. So these are the major things that happens at this stage, at stage 3. So you just need to know that at first, the spindle fibers attaches to the pair of chromosomes or the sister chromatides, and then it align them on the metaphase plate, or you can even talk about the kinetochore and or the centromere. This is what they are really interested about. And when we start looking at the checkpoints, I'm going to mention maybe some additional information that happens at this stage, but that's maybe when I finish explaining the stages, I'll talk about the checkpoints. Okay, I'll explain to you what checkpoints are and the checkpoints that are involved throughout this process. So now let's look at the fourth one. So the fourth one is called the anaphase. So at the anaphase, actually, this one, the metaphase, the name actually came from what happens here, the arrangement at the center. Meta, the word meta represents something that it represents like the middle point of something. Okay, so the name here, this word meta, it was because of this alignment of the chromosomes at the center. Okay, so let's talk about now the anaphase. So the anaphase, so when a cell just enters this phase, it will be looking like this. So it will be looking like this. We have the centrioles here, and the chromosomes or sister chromatides have been arranged. They have been aligned here at the center, like this. And then now, here's what happens. So now, when it just enters like this from the previous stage, at this point, it is where now they are going to be separated. It is where the sister chromatids are going to be separated. So now, how do the chromosomes get separated? So here's what happens. So when the chromosomes are attached here at this point, there is a protein that is called the CDC20 protein. It is because of this protein that there is an attachment here. Okay, there is an attachment here. So now, what happens is that, first of all, the chromosomes, the proteins that are here between the chromosomes that are attached, they begin losing the ability to hold the two chromosomes. In other words, you hear them saying they become, or they get cleaved. So they begin to lose the strength to hold now these two chromosomes here at the center. Okay, so now when that happens, the spindle fibers are going to start shortening, meaning that the length is going to start decreasing, which will now make now the chromosomes to get separated. Okay, so they start like going backwards. Even these that are on this side, they start like going backwards as they are shortening. Now, as they are moving backwards, remember, each spindle fiber is attached to a certain chromosome. Okay, so let's say maybe if they are like this, they'll eventually now start looking like this. Okay, when they were initially like this. Let's say these are the spindle fibers. So at first, they will be, here at the center. At first, they will be attached like this, and as they start shortening now, they start pulling away the sister chromatides. Okay, so now, eventually, they are going to be separated like this. So the chromosomes are going to be pulled to the opposite directions, which eventually makes now the chromatids to even separate. And one more thing, remember the nucleus here, which had a nuclear envelope that of course had to break down. It undergoes what is called karyokinesis, meaning that this nucleus here is going now to separate. So meaning that eventually you are going to end up with two nuclei. Okay, meaning that there is eventually now going to be one. They'll be looking like this. Okay, they're going to look like this because you are going to end up with two daughter cells, and each daughter cell need to have its own nucleus. So that's why it is at this stage now where the nucleus it kind of like gets divided into two. Okay, so that you can end up with two nuclei. So now at the end of this stage now, the cell is going to look like this. It is going to look like this. So you're going to have now these are the chromosomes. Actually, let me just do it like this. Okay, so now these are the chromosomes that have been separated. Okay, they have been separated, and then you have two nuclei. This is how it's going to look like now. They have been separated. Okay, here is there's just a single one here. Okay. Okay, so now two things happen here. The nucleus undergoes karyokinesis, or it divides, and then the pair of chromosomes, or the sister chromatids, are going to be pulled away from each other, or pulled to the opposite direction. Or in short, they are going to be separated. So separation and separation of the chromatids and karyokinesis is what happens at this stage. Are the major things that happens at this stage. Now let's look at the last stage. Okay, so now let's talk about the last stage, which is telophase. Okay, so this is where the cell is coming from the anaphase. It will be looking like something like this. So it will be looking like this. The nucleus has undergone karyokinesis, so it has split it. Okay, and then right now you have the chromosomes have been separated. Okay, they have been pulled to the opposite side. So now after that, this is how it's going to look when it enters this stage. So this is how it is going to look like. Now at this stage is where now the cell has to divide. Okay, so before it divides, first of all, remember that the nuclear envelope initially it broke down, right? So that the chromosomes can go out, can escape. Now at this stage, they reforms. Okay. Okay. So after that happens, now the cell will undergo what is called cytokinesis, written like this. Cytokinesis. There's a difference. At first, in the anaphase, it undergoes karyokinesis. Now, for cytokinesis, is where the cytoplasm has to split. Okay, with the division of the cytoplasm. So it's going to divide to end up with two daughter cells. Okay. So two daughter cells with each daughter cell. So the daughter cells are going to look like this. Now this time, each daughter cell will be having same number of chromosomes. Okay, this is like the nucleus. So we'll be having same number of chromosomes, as the parent cell. Now, say they might ask you now to explain how cytokinesis occurs in plants and animals. Okay, so in animals, cyto, I mean, okay, so they might ask you maybe to explain how cytokinesis occurs in the animal cells. So for the animal cells, so initially the cell will be looking like this. Okay, now it starts forming something that looks like this here at the middle of the cell. Let me draw it here. So it starts forming this shape between, okay, which eventually now causes the cell to split, and this formation that is formed between the cell is what is called the cleavage furrow. So the animal cells easily divide by just the formation of this cleavage furrow. It will eventually divide, okay, because it doesn't have any cell wall. Okay, there is no cell wall. They just a cell membrane. Okay, so that's why they have different ways in which they divide. Now for the plant cells, it is quite different, and this is how they undergo cytokinesis. So initially, the plant cell will be looking like this. So now, because of the presence of this cell wall here, they cannot be divided by the cleavage furrow formation easily. Okay, so the way that the plant cells divide is by, so there is something that's being formed here between. Okay, it just start developing slowly, and this is called the cell plate. So this is called a cell plate. So, and remember, the cell walls are made up of the cellulose, right? And the lignins, which even makes them very strong. So even this cell plate that is formed, it is also made up of some carbohydrates. So it is also strong. So as it grows, it eventually protrudes through now the cell, through the cell, the cell wall of the plant cell, which will now cause the plant cell to eventually divide, to eventually split. Okay, so without the formation of this cell plate, they wouldn't have been probably dividing. Okay, so it's the reason why the plants even divide. They cannot be. Okay, so now let's talk about the cell checkpoints. So remember that a cell, for it to, while it is undergoing the cell cycle, it passes through some stages, right? It can go from we said it starts from G0 at the interface, and then it goes now to G1, and then the S phase, then the G2, right? So now, and then from here it went to the prophase. Okay, so now when a cell is moving from one stage to another, there are some stages where it needs to be checked before it enters a certain stage. Okay, when it is moving from some stages going to another stage, it needs to be checked if it has met some conditions. Okay, so there are some conditions which should be met before it proceeds into the next stage. Okay, so these are what they call checkpoints. So it's like a cell needs to be checked if it has met certain conditions. If it hasn't made those conditions, then it won't be allowed to go in the next stage and proceed with cell division. Okay, it will maybe try to work on those conditions until they are met. That's when now it can proceed. Okay, so let's look at these checkpoints. Okay, so let me say now the checkpoints. So the first checkpoint, they might ask you maybe to mention them. There are actually three, and you need to kind of explain what happens on each checkpoint. So the first one is the G1 S phase. It is written like this, meaning that when it is moving from G1 phase to S phase, okay, here from G1 to S synthesis stage, I don't know if it is phase, where the DNA has to replicate. So when it is coming from this stage, going before it proceeds to this stage, it needs to be checked. That's what it means when you see that it is written like this: G1 to S checkpoint. When it is about to move or to enter this stage of S, it needs to be checked. So now, what is being checked at this stage? So remember, at G1 is where the cell has to grow, right? So that it can accommodate the materials that are going to be replicated. So that materials that will be, so it's at this stage where it has to grow, and also it is where it has to obtain some nutrients. Now, before it goes in the next stage, it needs to be checked if it has done what is supposed to be done at this stage. Has it grown? Because it needs to grow so that it can accommodate what is going to be replicated, and it needs some nutrients, because nutrients are needed during this whole process. Okay, so it will be checked when it is coming from the G1 and about to enter the S phase. Okay, so those are the major two things that are being checked. Okay, so you can say the growth is being checked, and you can say, you can just say the nutritional state. Okay, the nutritional state. Okay, so this is the first checkpoint that is found in the cell division. Now let's look at the second one. So the second one is called the G2 to M. This M is representing mitosis. Okay, mitosis is now when it is about to enter the prophase. That's when it is about to start now the real mitosis, the real cell division. So it's when it's coming from the G2 phase, which is the last sub stage on the interphase. Now it's about to enter the prophase. Okay, now remember what happens at the G2 phase A. So now remember, it's about to enter the process of mitosis, right? And I said that at the G2 phase is where some organelles are formed, right? There's most of the organelles, like the mitochondrion, the endoplasmic reticulum, like, let me just say all the cell structures, but let me just use the word organelle. So all the cell organelle are formed at the G2 phase. Okay, now, and then when it just comes from this checkpoint, remember that it passes through the synthesis stage A, right? And this is where the DNA has to be replicated. So even as it enters the M stage, which is the mitosis stage, the prophase, the DNA should be replicated. So some of the major two things that happens at this stage, the first one is that it needs to check for production or for formation of organelles, but let me just use the word production. Okay, so it checks for reproduction of the organelles, reproductions of the organelles. Okay, and then the second thing is that it need to check the DNA replication. Okay, and the DNA should be replicated. So if these are not met, it won't be allowed to proceed to mitosis. It won't enter the prophase. Okay, so for DNA replication. Okay, and maybe you can talk about DNA integrity. Some lecturers also talk about this one here. It's very important. It simply means that the DNA should not have any damages. Okay, it shouldn't be affected in any ways. The DNA that has been replicated, it shouldn't have any form of damages. So these conditions have to be met. Let's look at the third one. Okay, so the last one, the last checkpoint is called the spindle check. So this one occurs at the metaphase, at the metaphase stage, at the metaphase stage. So at this checkpoint, what happens is that remember, this is where the spindle fibers have to attach to the chromosomes, right? They have to attach to they need to attach to the chromosomes and then pull them away. Arrange them at the center, right? At the metaphase plate. So the first thing that is checked, it need to check if there is a correct attachment to the chromosomes. Okay, so it need to ensure that there is correct. They need to be correctly attached. They need to be correctly attached to the chromosomes. And not just that, they need to attach, because there are some scenarios where maybe some chromosomes haven't been attached with the spindle fibers, and that can lead to a condition. They ask about this question, so it can lead to a condition called, it's called aneuploidy. So aneuploidy, it is where there is improper attachment of the spindle fibers to the chromosomes, and there is no proper attachment. It can lead to the daughter cells not having same number of chromosomes. It can lead to the chromosomes not being distributed evenly or equally, because eventually both the daughter cells need to have the same number of chromosomes. Now, if maybe there might be cases whereby let's say there is no proper attachment here, it can result maybe the one daughter cell having more chromosomes than the other. Okay, and that will now lead to some condition called the Down syndrome, but they don't go that further. But for now, just know that it can lead to an aneuploidy, okay, which is due to the improper attachment of the spindle fibers to the chromosomes, and it causes an equal distribution of the chromosomes between the daughter cells. And then it also need to check that the chromosomes are arranged at the center, at the metaphase plate. Okay, they need to be arranged or aligned at the metaphase plate. Okay, so you can say that it checks if the chromosomes have been aligned at the metaphase plate. Okay, so this is all about mitosis. These are the very important things that you need to be familiar with, and make sure that you subscribe to the channel. And for more lessons like this, make sure that you join my Telegram group, where you find recorded video lessons, included meiosis, and other topics that you need to be familiar with. So this is Joseph here. Okay, so these are the groups where you are going to be added once you subscribe to the official group for A Plus Academy. As you can see, there is a group for MA110 there. In each group, I upload video lessons which are recorded. All these video lessons have been uploaded, and I still upload them. And in every lesson, I make sure that I solve tutorial questions, especially the most asked questions, so that you can learn and watch at your own pace and revise. Even physics video lessons are already there. Okay, and chemistry, everything is in the group, and the lessons are very organized. You'll be easily learning. Okay, so even biology, the lessons are already there. There are a lot of videos. You'll be just watching them at your own pace. And not just that, there is also this group for A Plus Academy solved documents. So this group you also be added once you subscribe. So this group is where I upload all the solved materials. Okay, even the documents that haven't been solved, starting from last year's past papers and tutorial sheets, I've solved them all. Remember, that's where the questions come from, and oftenly they repeat questions from the last academic year. So I've already solved all the documents. Even the tutorials that they'll be releasing, I'll be uploading them in the same group together with the assignments and the lab reports they all be uploaded in this group. So once you just subscribe, you'll be added in all these groups in just a single subscription. So make sure that you subscribe.